All right. In this video, we're going to be looking at Unit 5, Lesson 2, Exploration 1. We've been talking about cells and specifically the cell cycle, which is the process that cells go through in order to replicate. But a really important element of that division process is the cell's ability to package and divide up their genetic material. And so in this exploration, we're going to be looking at chromosomes. Now, we'll do an entire unit on DNA later this semester. But as far as just a brief introduction, you're probably familiar with DNA, at least a little bit. DNA is the genetic material that contains information for how an organism's characteristics are inherited. Later on, we'll talk about how DNA provides all of the instructions for how to make every single protein that your body needs, which is a big deal. But for each new cell that is created, it has to have the complete set of DNA available so that it knows how to function properly. And so before that division process happens, we also have to be able to replicate that DNA, which again, we'll talk a lot more about how that happens in our unit on DNA. But I want to introduce this topic to you because it's going to be important in understanding how the cell can go about this division. When you look at DNA and chromosomes, it's important to understand some terminology. So, first of all, a chromosome is just one long continuous piece of DNA. It's almost like a scroll. If you think about back in the day when information, books, historical events, they weren't recorded on paper like we have now, bound by books. They were recorded on long scrolls, and just like you might have multiple chapters or multiple volumes of a history. In humans, at least, we have 46 different pieces of DNA that make up our genetic code. Every organism has a different number of chromosomes, but in humans, that number is 46. And again, we'll talk a lot more about how those chromosomes are organized. We'll talk a lot more about what they mean later on. But just in terms of terminology goes, it's important that you know what a chromosome is. Chromosome is just a piece of DNA. Now, that piece of DNA, if you were to pull it out and lay it end to end, it would be about 10 feet long, which is crazy because 10 feet worth of DNA in every single one of your multiple trillions of cells would get pretty long. So how do we organize it? Well, again, this is something we'll talk a lot about later, but for the structure of our discussion today, this DNA molecule is twisted and coiled multiple different times in multiple different ways until it is eventually formed into what we kind of call an X. Okay, it kind of has this X shape to it. And again, this X shape is going to be representative of an entire chromosome that contains one set of information. So you're actually going to have 46 of these X-shaped structures in your DNA code every time it divides. So let's talk about how that happens. Now, for the interphase portion, which if you remember, interphase makes up the large majority of a cell's life. During interphase, that DNA is not coiled up. It's what we call loose. And the reason for that is in order for it to be used, it has to be loose. Like I mentioned a scroll earlier, or you can also think about a book. In order for you to be able to read any of the information on a scroll, it has to be unrolled. In order for you to be able to read any information in a book, it has to be opened. And so DNA is the same way. That in order for that DNA to be able to be read, it has to be loose. It can't be packaged up. It can't be coiled, because otherwise you wouldn't be able to read the information inside of it. Now again, all of this, as far as how your cells read the information, that will happen later. But for now, just know that during interphase, that DNA is in a loose form, and we call that loose form chromatin. Now, for some reason, scientists, when they named all these things, they chose to use like the most confusing system possible because one letter is going to separate the difference between two pretty important ideas. But chromatin with an N is the loose form of DNA. This chromatin is how DNA exists for most of the cell's life, but at some point when the cell decides it's time to divide, that chromatin is going to go through that condensation process where it's going to get tighter and tighter and tighter, like this picture, until eventually it's going to form a chromatid. Now, this picture, where it looks like an X, is actually two chromatids stuck together. A single chromatid is going to copy itself during interphase, and then those two copies that are identical to each other are going to stick together to form this X-shaped structure. So again, chromatin, loose form of DNA, chromatid with a D, condensed form of DNA, and then sister chromatids are when we have those two chromatid pieces that are identical to each other stuck together to make that X-shaped. Here's an electron micrograph picture of what this actually looks like. It's pretty cool. You can see the X-shaped here. So this is actually represents two copies of the same chromosome that has been condensed into that chromatid form. And again, when they stick together to form that X, we call them sister chromatids. Now again, in order for the cell to be able to divide, it's going to need to accurately and really perfectly separate all of those chromatids to where each new cell eventually contains one complete copy of the total volume of DNA. So in humans, what that means is that there's going to be a process that kind of evenly divides out all 46 copies of the chromosomes, 46 on one side, 46 on the other side, so that both new cells are able to have a full set of DNA. And that's what we're going to focus in on this lesson, how that actually happens. There's an interesting example at the bottom of this exploration that I do want to talk about briefly. One of the ideas behind aging is that every time your DNA is copied, there's a small section of DNA that is cut off called a telomere. At the end of your chromosomes, each one of your chromosomes has this extremely long stretch of letters that doesn't mean anything. It literally just is this telomere tail. But every time your cells divide, a small portion of that tail gets cut off. And some scientists have suggested that this is almost like a countdown. That as that telomere gets shorter and shorter and shorter and shorter, that effectively is what amounts to the aging process that we experience until eventually our telomeres get so short that they can't function, our cells begin to break down, our DNA can't copy itself, and that's ultimately, one of the things that leads to us dying. There's a lobster, and lobsters in general, they don't appear to die from old age. They die from other things, but they don't die from old age because their telomeres don't shorten. And so one of the areas that humans have looked into is how can we possibly harness this process of not shortening telomeres to allow us to live longer. There's an evolutionary scientist named Brett Weinstein who's got a pretty good thought on this. But there's a tricky balance there because one of the benefits to telomere shortening is that it prevents those cells from becoming cancerous. It's kind of like a fail-safe thing, because if a cancerous cell decides to start multiplying rapidly, one of the fail-safe mechanisms is that they burn through their telomeres so fast that they eventually go away and can't cause any more problems. And so, in this area of research, there's a really kind of important balance that people are trying to find out, which is how do we keep telomeres from shortening to the point of aging, while at the same time building in fail-safe mechanisms so that when those cells become faulty, which they do, that they don't lead to cancer and a bunch of other terrible things happening. They've studied this in mice a lot. And the problem they found is that the mice who don't die from aging almost always die from developing tumors and cancer. And so there's yet to be a solution that allows for the aging process to be slowed down without at the same time making the likelihood of cancer much higher. So an area to look for, maybe sometime in your lifetimes you'll be able to see this solution looked at. But there's an interesting thing here to read about these lobsters, because they open a door to a potential solution that we've yet been able to walk through, but might be something someday. So, all right, that's it.